Hello, my name is Bridget, and this is my partner, Henry.
My name is Henry.
And today we'll be discussing our presentation over the exploration of biological hallmarks of cancer in musculus cell lines, A9 and mutated PA28γ deficient cancer clones. Cancer is a series of genetic diseases caused by an accumulation of mutations resulting from a genetic predisposition and environmental factors. As cancer is the leading cause of death worldwide, understanding the biological capabilities that compose the hallmarks of cancer could provide an important avenue for treatment and prevention. In this study, the presence of these biological capabilities are evaluated in PA28γ deficient cancer clones in comparison to tumorigenic cells of known phenotype in PA28γ deficient mouse embryonic fibroblasts. PA28γ is part of the 11S regulatory protocol and activates the 20S proteasome. In addition, it is important because previous research has indicated its importance in cell cycle regulation and proliferation control, as well as the centrosome and chromosomal stability maintenance. In this experiment, we hypothesized aneuploidy and genomic instability will be the highest in KOCC cells, that KOCC cells will be the most migratory and invasive, while tumorigenic A9s will be the most proliferative, and KOMEF cells will be the most resistant to treatment by Cladribine and Taxol. In figure 1, you can see our project overview for the semester. In figure 2, you can see how karyotyping revealed KOMEF cells to have the highest aneuploidy and how A9 and KOCC cells had the same percent aneuploidy. Figure 3 shows us how flow cytometry data was not indicative of the expected range as exemplified in part B. This makes us rely more heavily on karyotyping to draw conclusions on genomic instability. Figure 4 exhibits how KOMEF cells had the highest proliferation as an aspect of wound healing in a wound healing assay. KOCC had the highest percent of invasive cells as a characteristic of invasion.
All right. In figure 5, A9 cells are more migratory than KOCC cells, and KOMEF cells show no invasive capability. In part A of the figure, representative images of the A9 and KOCC transport assay demonstrated that the cells from both cell lines were able to migrate between the pores of the filter. And part B shows the percent migration at 24 hours, which was determined that the A9 cells were the most migratory. In figure 6, we can see that A9 cells are more resistant to treatment with Cladribine and Taxol than the KOMEF and KOCC cells. Part A shows flow cytometry histogram showing viable, apoptotic, and dead cells. Part B shows the summary values for the flow cytometry and percentages, and part C is the MTS assay examines mitochondrial activity of A9, KOCC, and KOMEF to determine cell viability, which shows A9 to overall be the most viable in these conditions. In our conclusion, we see that as a cancer clone of PA28γ deficient, a PA28γ is a regulatory of proliferation and other nuclear functions. Its reduced capacity for carcinogenic hallmarks may be a result of this loss. We see that A9 exhibit approximately 40% more migratory ability than KOCC. KOCC and A9 exhibit the same aneuploidy. Tumorigenic A9 cells were more migratory and invasive, as well as more resistant to anti-cancer treatment drugs like Taxol or Cladribine. In our future directions, we would like to determine the IC50 of the chemotherapeutic treatments, explore possible mutation in PTEN, HRAS, and P53, and replete our flow cytometry technique in order to detect the levels of genomic instability of different cell lines. We would like to thank the Discovery Foundation for Omega Training Support Program, and the AC Biology Department for their financial support for this project, as well as the Cancer Bio classes of 2019 and 2021 for their collaborations. Thank you.
Thank you.